Okay, welcome everybody to BSI Webinar Winter 2013 2A, which is Foundations of Neuronal Dynamics and Live Z-Score Training with Dr. Tom Collura. If you are here for CE, pay attention for the validation statement that'll occur somewhere in the middle. If you have any questions or having any technical issues, feel free to call us as soon as possible. Everyone is going to be muted, and if you have a burning question, you can type it into your chat box, but usually we wait until the end for any questions. And now I leave it to Dr. Collura.
Okay, thank you, Cindy. I'm assuming everyone can hear me. I'd like to welcome everyone. Thank you for your time and your attention. I understand we have a little over an hour to go over the material, and then there'll be some time for questions. So I see the screen is alive and flipping. Okay, so welcome to the Webinar 2A, Foundations of Neuronal Dynamics and Live Z-Score Training. I'm going to be presenting material that's been developed over the last about 7 years or so. We have a lot of data to share and a lot of theory to talk about. So without further ado, we'll begin the presentation. Just for the record, right now I am with Brainmaster Technologies, and I am also a instructor with Stress Therapy Solutions. And I am also with the Brain Enrichment Center, which is a clinic in Bedford, Ohio. And I am completing my master's right now in counseling, in mental health counseling. So we are putting this material into direct practice as we speak. And it's very exciting to be involved now in actually applying these techniques in addition to developing them. We always harken back to the earliest studies. This would be 1924 when Hans Berger began to try to record scalp EEG. And he actually had to use needles and other invasive things and a galvanometer because there were no amplifiers and no transistors and not even tubes in those days. So after a long period of time, almost 10 years of work, he was able to produce the first EEG recordings. And what I want folks to notice in this particular tracing, which is shown at two different scales, the smaller scale on the top and the larger scale in the middle, and on the bottom is a 10 cycle per second rhythm generated by an oscillating mirror, that Berger was not only the father of EEG when he took this recording, but he made several observations which actually make him the father of QEEG. And those observations were that the EEG is built up of a mixture of frequency components. And he immediately recognized a 10 cycle per second component, which is clearly visible, especially in the top trace. And he called it alpha. And then he recognized a faster rhythm, which is more evident in the middle trace, which is about 20 cycles per second, and he called that beta. And he articulated the fact that the EEG is composed of a mixture of these frequencies as a complex kind of harmonic wave. He further speculated that one could apply mathematical techniques to discern these frequency components. And he even mentioned the Fourier transform as one mathematical technique to reduce a signal into frequency rhythms. So in making this observation, he really anticipated the use of modern frequency analysis in the analysis of EEG. And that sort of makes him the father of QEEG, as a matter of fact. Later on, shortly thereafter, other investigators began to identify delta waves, which are the very slow changes in the baseline. And they also began to recognize the theta waves, which are visible as sort of a non-sinusoidal low-frequency oscillation, also in these very rhythms. So very, very early on, we had delta, theta, alpha, beta, and the concept of frequencies in EEG. Everything we're going to talk about today builds right on top of that very, very basic structure. Now, what Berger was trying to do, and what he ultimately succeeded in doing, was by taking electrodes and putting them on the scalp, around the skull, or actually he was using needles which penetrated the skull. He realized he was measuring electrical activity that was produced by internal structures. And although they did not have quite the extent of the neuron doctrine that we have today, from Eccles and Lorente Deneau and Ramon y Cajal and these other great pioneers, he nevertheless believed that he was measuring electrical activity. And we know now that the brain sources produce what are called dipoles, which are little tiny regions of positive and negative activity, and they produce spreading fields, and those fields can be read from the surface of the scalp. And part of our goal then is, by using these surface measurements, to ascertain something useful about what's going on inside. And we will refer to the dipole theory as we describe and explain what people are seeing in EEG. So at a deeper level, we know that the cortex consists of layers, and that these layers contain a variety of cells. And the cells that are most responsible for EEG happen to be the pyramidal cells. And we see pyramidal cells marked with the letter P in this drawing. And they produce significant postsynaptic potentials across their membranes. And because they're oriented like little soldiers in a row, it's like everyone's facing the same direction, we're able then to ascertain that when they work synchronously, when you have large populations of pyramidal cells depolarizing synchronously, now you get measurable potentials that can be measured from the surface. So even as we move into z-scores and mathematical analysis, we're always thinking in terms of what are the underlying brain structures, what are they doing, and how does it relate to the EEG we're measuring. One of the most important qualities that we see in the EEG anatomy is that this connection between the thalamus, which is in the brain, it's the top of the brain stem, and that's the region in the middle of this drawing, and how it relates to the cortex, which is the areas all around the circle of this drawing. So the thalamus can sort of be thought of as the peach pit in the middle, and it has projections all around to the surface. And virtually all the rhythms that we see are intimately involved with thalamocortical activity. Now, the slower rhythms tend to be more subthalamic in nature, and the faster rhythms tend to involve thalamocortical projections, such as to the motor strip, visual cortex, and such. And the faster rhythms are actually cortically generated. So you can picture, and there's some drawn here, connections from one area of the cortex to another area of the cortex. These are what are generating these rhythms that we're seeing. And what we're going to find out is, even as we measure from the scalp, the electrical potentials primarily arising from the cortex. It's the thalamocortical reverberation and the total brain systematic activity that we're looking at when we look at rhythms, waxing and waning, speeding up, slowing down. So an important thing to know is that a primary mechanism of the brain is inhibition. If we go back to the drawing before, I'll mention that the nuclei which sort of surround the thalamus are almost entirely inhibitory. Their main job is to hold things back. And that's also true of certain areas of the brain, such as the frontal lobes. They perform a very inhibitory influence on the rest of the brain. And we tend to see rhythms when the inhibition is relaxed. So the generation of a rhythm can be thought, rather than actually producing something, more like allowing something to occur. It's when inhibition lets go that rhythms occur. And in fact, an extreme breakdown of inhibition is what is known as epilepsy, epileptic seizures. So when you do have a normal amount of inhibition and it's being relaxed, you can have thalamocortical relaxation, thalamocortical reverberation. And so the cortex and the thalamus are kind of releasing their inhibition and allowing rhythms to occur. And then the populations can oscillate in synchrony. And it's when populations are synchronously active that you're going to see the brain rhythms, such as typically theta, alpha, beta, the mid-range rhythms. Now here's a picture that we generated when I was back in the surgical world, back at the Cleveland Clinic working with epilepsy. And what we have on the left is a scalp measurement of a movement-related potential. And you can see that those scalp waves, these are evoked potentials, you can see that they're spread all over the brain. It's maximum at C3, you see that deflection. And then you also see significant deflections all over, C3, C4, even the frontal areas, posterior, parietal. That's very, very spread wave. On the right-hand side, we have the invasive recording that was made with sensors put directly on the surface of the brain. And those sensors are just about a centimeter apart. And what we can see is that while some of the sensor locations are extremely active, give very high activity, the area right next door to it might be very, very low or basically silent with respect to that. So there's an enormous amount of cortical specificity and localization inside the brain, invasively. It's telling us that the dipoles are highly localized and very isolated. But on the scalp, it's going to be spread. And you're going to get a distributed scalp potential. And EEGers know how to read this, and they know how to recognize these. One of the reasons Z-scores are so important and valuable is that they take into account this spreading. So when you see Z-scores on the scalp, the software knows what those normal amplitudes should be in all different scalp locations. And use of Z-scores allows us to make very accurate maps, and even, as we'll see, to do neurofeedback that's very scientifically based on the underlying activity. So as a sample for an EEG, you might say, okay, there's a generator down in some particular region. This would be, for example, of kind of a mesial temporal location. And the activity spreads electromagnetically. Basically, it's volume conducted through the fluid of the brain. And it makes its way to the surface, and it winds up on an amplifier that we put there strategically. And that's what gives us our EEG voltages that we're measuring. Now, when we think about the brain now, this kind of relates to the drawing I had before. On the bottom, we would have the brain stem structures, and then up above that, we have subthalamic structures, inferior colliculus, superior colliculus, nucleus accumbens, globus pallidus. Those participate a lot in theta, lower rhythms. And then in the middle with the red boxes, we see the thalamus. And the thalamus has these profuse projections all to the cortex, and the cortex is the box up on top. And in the cortex, we see auditory, visual, somatosensory, motor, prefrontal, limbic, and cingulate cortex, all with projections from the thalamus. All these different nuclei in the thalamus project up to the cortex. And depending on those projections, those are producing the rhythms that we're seeing. For example, alpha 2, the 10 to 12 hertz kind of alpha, is mediated by the lateral geniculate projection to the visual cortex. The slower alpha, what we call alpha 1, 8 to 10 hertz, is primarily seen frontally, so it's related to this medial dorsal projection to the prefrontal cortex. And we're going to hear a lot about Barry Sterman and SMR today. And we now know that the SMR rhythm is generated by the projection between the ventrolateral thalamus and the motor cortex, C3, C4 regions. So all the rhythms that we're seeing basically are due to either thalamocortical types of interactions or cortical-cortical interactions. All the blue lines up on top are also generating rhythms. So you can think of this as a very complex kind of an engine, signal processing engine, and we're trying to kind of see what's going on under the hood by measuring these rhythms. So there's a natural cycling process between excitatory and inhibitory neurons. And it's fairly clear seeing this, that excitatory neurons, they increase their activity, then the inhibitory neurons are stimulated, and they begin to act, and then they begin to inhibit the excitatory neurons. So there's a cyclic reverberation going up and down and up and down. And this needs to be flexible and cyclic and rhythmic in order for the brain to function properly. If this is stuck at any point in the cycle, you're going to have problems. If it's excessive, if there's too much cycling, sort of out of control, now you're going to have other problems. And the problems are going to depend on the location where the irregularities occur. It's not so much the frequency that we're worried about, it's the location where there's a dysregulation. And again, Z-scores allow us to get a handle on what is the normal amount of rhythmic activity, what is the normal amount of any frequency at a particular location, based on this kind of a model, is like how I like to think of it. Is the system exploring its dynamic range? Is it flexible? Is it healthy? Now, Barry was one of the great contributions he did for us, and we owe him an enormous debt of gratitude, is he did demonstrate in his work with David Kaiser rhythmic cycle which occurs in normal good performance, and they found it in pilots. And they found that effective pilots cycled naturally between a beta type state and an alpha type state. And the brain would go back and forth and back and forth. But they were in appropriate states at appropriate times. So basically it was found that good pilots precede a given task with a high frequency, low amplitude EEG, a state of readiness. And then after the task was completed, their brain would produce a low frequency, high amplitude EEG, basically alpha, and he called that PRS, post-reinforcement synchronization. The poorer pilots did not exhibit this type of cyclic activity that seemed to be quite under some sort of unconscious control. And as a result, the poorer pilots, when they needed to perform a task, they might be in alpha. And then they would have to go into an activated state. And that took more time, which reduced the reaction time. They also did not do as well because they were not really ready in time, and they got fatigued earlier. So the whole concept of good performance and mental efficiency is intimately bound with the normal, natural, rhythmic cycling of the brain between a readiness or concentrated state and a relaxed state. And you need both. You don't want to be stuck in one or the other. And a brain that is healthy and flexible is going to move between these states with ease and with efficiency. So I can show these states sort of on a continuum. And on the left side, we have low frequency, high amplitude activity where the brain cells are very synchronized. They're less independent. They're more dependent on each other. And this is a particular state. At an extreme, it would be a theta state with very high amplitudes. And as you move to the right, you go through an alpha state, through the low beta state, up to a high beta state, which is mediated by very fast cortical connections. They are low amplitude rhythms. They're higher in frequency. There is less synchrony, but there is more neuronal independence. So any system tends to want to move between these. And this is not just brains. If you think about it, you'll realize that the body itself, communities, social systems, even people's entire lives need to move back and forth between these states, because staying too much in the relaxed synchronous state is not going to give you enough information processing, enough ability to change. Being stuck on the other side, where everything is less synchronous and independent, is not going to allow you to rally resources together. So it's a very basic idea. And again, I look to Barry as one of the first people who really articulated this and showed how it relates to mental efficiency. So in addition to this activation issue, we're also interested in connectivity. And connectivity then has to do with how much things are connected. And we have, for example, coherence, which is a measure of the amount of shared information between sites. We look at phase, which is a measure of the speed of shared information between sites. And this whole concept of connectivity exists in many dimensions, but one primarily is thalamo-cortical. What is the connectivity between the thalamus and the cortex? And that connectivity helps give rise to theta, alpha, and SMR type rhythms. And we also then have cortical-cortical connectivity, which is important. And those are in the higher frequencies, beta and gamma. And for example, an intrahemispheric connectivity, let's say in the left hemisphere, is going to be very important for language. And you're going to need normal amounts of connectivity in order to have language function, whether it's reading, writing, speaking, planning, speaking, these types of things. We also have interhemispheric connectivity, very important. For example, in the frontal areas, the connectivity between the left and right frontal areas is very related to attention and control. For example, we know that in autistic kids, Rob Coben and others have pointed out that there would be possibly connectivity abnormalities, either high or low, particularly at lower frequencies. We also have an important aspect of occipito-parietal connectivity. Are the visual areas connected with the association areas? So is sensation connected with perception? And these are important for sensory integration. And I'll also mention that the occipito-parietal connections are among the first to go in aging. So when people start to lose certain types of sensory binding capabilities, and you can imagine where it shows up in driving ability. Because in order to drive, you need to be able to see the cars around you, you need to be able to figure out where they are, what they're doing, where they are, and you actually need to integrate color information in order to drive. And our work with Brown University, we found, for example, that it's specifically the integration of motion with color that is related to that occipital-parietal connectivity and can actually be dealt with using not only QEEG, but Z-score training to help develop those connectivities. And I'll show some examples of that. So the coherence, just to make sure people are clear, it reflects the similarity of activity between two channels. It's a measure of the information sharing. Coherence can be trained up or down. And people who are not using Z-score training typically have to work with these decisions and decide if they're training it up or down. What we're going to see is when you use Z-scores, you don't have to worry so much about it because we can range train it. And our friend David Kaiser has articulated what he calls the Goldilocks effect, that the coherence may be too high or too low, the same way that Goldilocks found that the bed was too hard or too soft, or the soup was too hot or too cold. Coherence needs to be just right in order for the brain to function. Too much coherence means you're too tightly connected, there's not enough differentiation. Too little coherence means there's not enough communication. Things aren't working together. One interesting sort of exception is that alpha coherence, we pretty much find you can train that up in the back as a peak performance type of training, and very little adverse reaction is found there unless you really have an abnormal situation. Certain types of people are going to not like alpha training. But typically people like James Hart, Les Femi, Adam Crane have been training alpha coherence up for decades as a peak performance and a self-regulation thing. So we talked about amplitude as a metric. We talked a little bit about frequency as a metric. There are many ways to measure EEG. EEGs are as complicated as anything else, and like the proverbial elephant in front of the wise men, there's a lot of different ways to perceive it and to look at it. So we do look at amplitude, we can look at frequency, we can look at peak frequency, or we can look at modal frequency, which is an average frequency. We also look at percent energy, which some people call relative power, another important metric. We can look at variability, another important metric. How variable is the EEG? We can look at the coherence between two channels. We can express it as a percentage. We can look at phase. We can express that as in degrees of phase or as a percentage. We can look at asymmetry between two channels as a ratio or a percentage. Now, what I'll mention is kind of a heads up. Now that we have advanced localization techniques like Loretta and S-Loretta, these concepts can be extended not just to channels on the surface, but to regions of interest in the brain. And we'll be talking a little bit, and in a future webcast, I'm going to talk in detail about using Loretta, S-Loretta specifically, and measuring the communication between, say, the anterior cingulate and the prefrontal gyrus, or between the dorsolateral frontal cortex areas. So the Z-score training extends very, very naturally and very well into in what I call invasive localization work. Goes beyond the scalp and goes right inside quite well. So what about Z-scores? What's the purpose of Z-scores? And I'll mention that there's some misunderstanding out there. There's some concern that might not be as warranted. We take a very low impact approach towards Z-scores, and we put a lot of brain flexibility to work here. So to us, Z-scores are a method to understand a population. They are a method to understand an individual. It uses statistics to evaluate quantities. It's a standard method applicable to any measurement. It's very important for connectivity, phase, and asymmetry measures. And you may have mentioned I have not anywhere said that a Z-score of 0 is perfect or that everybody should be in the middle of a distribution. We do not believe that at all. We never did believe it. And I'm going to show some very interesting results where Z-scores are useful even when you're not in the middle of the population. You're not at 0 Z-score. In fact, nobody is ever at 0 Z-score. You don't want to be there. And I'll explain that why. So here's a normal distribution. We have males on the left, we have females on the right, and we're using height as a metric. And the metric could just as easily be alpha peak frequency or amplitude or beta coherence. And what we do in order to get a distribution is we line the samples up, the individuals up, and we take, for example, the men on the left, we have the short ones on the left and the tall ones on the right side of their bleachers. And everybody of the same height is standing on top of each other's shoulders, so to speak, so they form a histogram, a bell curve. So the men have a mean height, which is probably about 5'10" or 11, and a standard deviation, which might be a couple inches. So as you move to the left, you have negative numbers, and as you move to the right, you have positive numbers as z-scores. And on the right side, we show the females, who are also the athletes, and they lined up according to the same rule, but of course they produce a different mean, and they produce a different standard deviation, because there's physiological differences underlying those differences. So once we have a normal distribution, we can now convert any measurement into a z-score. And this is really all we're doing. We're not trying to put value judgments on anything, because clearly all of these athletes are normal. The short guys are normal. The tall guys are normal. By definition, the entire bell curve is normal. So no one is going to argue that the guys in the middle are more normal or better fit or anything like that. It's just a measure of their personal qualities. So there's your normal distribution. This is how we think of it. Reduce any number to a z-score. And 0 is the middle, as we've been saying. One standard deviation, plus or minus, takes in about 68% of the population. So 2/3 of the population is within one standard deviation plus or minus. When you go to 2 standard deviations, now you have more like 97% of the population. And as you go to 3 standard deviations, you now have 99.9% of the population. So it's relatively rare to see people at extreme z-scores, but by definition, in our work with our populations, we're using the databases that are built around normals. So we use the word normal distribution not because they're normal, but because that's the mathematical term. But we also happen to use references which are built around normal people. Now, there's no reason you can't do otherwise. And we are actively developing a database of college students and high performers. We've evaluated some high-performing individuals, and I will say that they definitely do not lie in the middle of these distributions. You could have a very high-performing individual with fast alpha, and it's going to show up with extreme z-scores, but it doesn't mean there's a problem there. And we certainly would not be trying to train them back to 0 for any reason. So the z-score is a measure. It's based on the current data value and the population statistics you're working from. All it tells you is how many standard deviations are we from the mean. It's defined as the measurement minus the mean divided by the standard deviation, and it's really nothing more than that. It's simply a normalized way of looking at any number, provided you have a reference. So, as I said, plus and minus 1, we use the word sigma instead of standard deviation because we save a little time. Plus or minus 1 sigma is 68%. Plus or minus 2 sigma is 95%. Plus and minus 3 sigma is 99.8%. Plus and minus 4 sigma is a very, very extreme. If you see someone whose z-scores are 4 sigma out, what you're going to say is not that they are an extreme example of normal, but it is much more likely that they actually come from a different population. So, for example, the database does not include a bunch of kids with ADHD. And the database also does not include a whole bunch of depressed individuals. So when you have a client who might have ADHD or depression or any of a large number of dysregulations, disorders, you're going to be looking for extreme z-scores. And when you see them, you want to associate them with the dysregulation. You want to associate it with the problem. I do not recommend that people do a knee-jerk reaction and see an extreme z-score and immediately try and worry about it. You need to understand why the deviation is there, what it means, before you try and do anything at all with it. So in a simple example of adult height, mean height is 6 feet, standard deviation is 3 inches, so someone who's 6 feet 6 is a 2. Someone who's 5 feet 9 is a -1. and someone who's 5 feet is a -4. Now, you'll notice that I said that the z-score of -4 is about 1 in 10,000. And in my life, I've probably met a lot of people, and I know exactly 2 people I've met who are otherwise normal but are 5 feet tall as adults. So I think that's kind of about the right answer, that you can have those very rare individuals who are entirely normal but short. But when you do see someone who's extremely short, it's more, and they're an adult, you more likely you're going to want to look for something else as to why is this person so short. Is there a physical deformity? Is there a nutritional problem? Maybe they're from an unusual population, such as very short pygmies, for example. Now, this shows where the z-scores come from. On the left, I've shown raw values from 2 individuals, and these would, for example, be alpha amplitude values. And the waves on the left are not the EEG waves, they are the size of the EEG. Think of it as the envelope or the amplitude values. And what I'm showing on the left would be about 2 minutes of time. So over that 2 minutes of time, you see one person with their alpha going up and down on the top, and another person with somewhat less alpha, again, waxing and waning, going up and down on the bottom trace. When we do a normal QEEG, what we look at is the mean over all that stretch of time. And that's the first pair of bell curves on the right. I show the ranges of the individual scores, and I show the means of those individuals. Now, when you build a database, you typically are going to take those population means and look at their distribution. And when you do that, you get the middle bell curve, the one that says range of population means. That is the bell curve of your normative database, whether it's NX-Link, BrainDX, NeuroGuide, Skill, whatever. Typically, you're looking at these population means. Now, when we work with live z-scores, we're actually looking at dynamic values. So you need to think more about the bell curve on the right, which is the distribution of values normally distributed throughout the activity of the individual. And one way I think of it is like body temperature. Everybody's body temperature at rest is pretty much the same. It's all going to be clustered within a degree or two of 98.6. But your body temperature actually goes up and down. It goes down when you go to sleep. It goes up when you're exercising. It can go up there in other circumstances. So the dynamic range of your body temperature is going to be much greater than that normal static range. And it's also affected by, obviously, fever. You might be hypothermic. So when we work with live z-scores, we're typically thinking of the dynamic range of these values, and it's a much wider range of values we can see. And as a result, we typically are looking at z-scores that have lower numbers because a given deviation is going to be less significant in that dynamic bell curve. So here's the approach we take. We compute ongoing z-scores. We apply them as training variables, and there's a lot of thought that goes into how we do that. We establish targets and criteria. They are by no means fixed at the outset. They are by no means everybody has to be 0. These targets and criteria, I'll be explaining these. We then provide feedback of various types for various reasons. And we do use predefined bands. They have to be unique and predefined and not adjustable because they're built on a population. And these bands are independent of those that might be used in the main software. So you could still be training, some crazy band like 12 to 20 or, 4 to 14. And still using z-scores in the fixed bands in order to monitor progress, see what's happening, add additional feedback, for example. So we take a very flexible approach. We typically now are using absolute power, as I talked about. We're using relative power, which is sort of percent energy. We use power ratios, which is the ratio of power between bands. We look at asymmetry, we look at coherence, and we look at phase. So if you have 2 channels, now you have, let's say we have 8 bands. So for each site, you have 8 powers, 8 relative power, 8 absolute power. You have 10 power ratios. And for the connection, you have 8 asymmetries, 8 coherences, and 8 phases. So all of a sudden, you're looking at a screen like this. And this is essentially a live QEEG report. And so what you see for two channels, we see F3 and F4. And we're seeing in delta, theta, alpha, beta, beta 1, beta 2, beta 3, what was called gamma at that time in this graph. We now actually go up to 50 hertz for gamma. You see absolute power with ABS over it. You see relative power with REL over it. These are all z-scores now. So you're seeing how far they deviate from that normal population. We also have power ratios as z-scores. And clearly the system it's what's called over-specified. There's a lot of redundancy in here, and it's admitted, and it's not a problem to have redundancy. We're just looking at these amplitudes and ratios in a variety of different ways. For example, you can see that for F4, the ratio to gamma of beta to gamma is a little low. It's minus 1.1. Well, you wouldn't see that just looking at the numbers for beta and gamma. You wouldn't even know what it should be. But now you know in terms of z-scores, there is a little bit of a deficit of beta relative to gamma. And then on the bottom, we show the pathway. And we show asymmetry, coherence, and phase between those two sites, between F3 and F4. And you see it again for delta, theta, alpha, beta, beta 1, beta 2, beta 3, and gamma. And we see in this case the asymmetry is relatively normal. We see a deficit of coherence in a lot of frequencies, which is going to be very interesting between F3 and F4. If you have a coherence deficit, that means they're not talking as much as they should. And we also have an excess phase difference, and that tells us that there's slowness between them. So just by looking at this, you can instantly see that between those two sites, the powers are relatively normal, absolute relative. There's not a power issue, but there's definitely a coherence issue. There's a connectivity issue, and there's a phase concern. This can be incorporated into training immediately, and it's very useful. There's no reason you might not want to use these scores to train this coherence back to normal, but not overtrain it, and train phase at the same time. In fact, we've even had clients who look at this screen and use it for training. I've seen clients sitting there staring at a phase number and saying, watch me make it go down. And sure enough, they'll drop it 2, 3, or 4 tenths of a point in a few seconds. So it's a very powerful technique. Now, when you go to 4 channels, it's kind of like the difference between standing on 2 legs and standing on 4. There's a lot more information in 4. Because for each site, you still have absolute power, relative power, and power ratios. But for the connection, you now have 6 pathways. There's 6 ways to connect 4 things. So for all the asymmetry, coherence, and phase, you now have 6 entries. So here's what a 4-channel screen looks like. Now, when we first started looking at this, some people thought, oh my gosh, look at all that information. The brain can't handle it. You know, I can't handle it. Nonsense. This is a very easy to read screen. You get very used to it very quickly. In fact, you know, we just rely on this screen. 4-channel z-score training is one of the most efficient, straightforward ways to get a lot of information about the brain. And look at this person. Over in relative power, we see these little excesses of beta 1. You see a 1.1 over there on the left for beta 1 under relative power. On the right side, we see a 1.4 for beta 1. That's just a little bit of excess SMR. I don't see that as a problem. It depends on the individual. But, I mean, as Barry showed us, basically, and as a lot of recent research has shown over decades, having a little excess SMR is not a bad thing. It indicates the ability to sit still. It indicates some relaxation, some mental integration. Unless you have a problem that you think is associated with that, this excess SMR to me is just not a problem at all. It's something we see as a signature. It's a personal signature, if you will. But when we go down to these connectivities on the bottom, you see 6 sets of numbers. F3, F4, again, we're seeing this deficit of coherence in the excess phase. For F3 to P3, that law looks normal. So that's telling us there's no left hemisphere connectivity problems. We're not likely to be looking at language problems, this type of thing. Now, the 2 middle ones are the cross connections, F3 to P4, F4 to P3. Those all look normal. I will mention, because there are no native pathways between F3 and P4 or between F4 and P3, deficits or excesses you see here tend to be global. It tends to be sensitive to what's going on more globally. F4 to P4 is normal again, so now we don't anticipate any spatial reasoning or spatial memory problems, anything associated with that. But P3 to P4, we see again a coherence deficit and a phase excess. Well, to cut to the quick, this points to some issue with the corpus callosum. There is an issue with cross-cortical connectivity. So we might be looking for attention problems. We might be looking for some kinds of verbal expression problems having to do with emotion. We might look for issues of integrating spatial and language information. And if we have concerns of those types, then training these coherences up and those phases down using z-scores is probably going to be a very reasonable thing to do. So we have z-scores, which is really just another form of targeting. It's just another way of saying how close are we to some target, and we give it in z-scores. We can train them up or down, simply directional. You can train them using a range. You can say, I have a range I need them to be in. And this is what most of the other providers are doing. They're training them into a range and basically trying to train all the z-scores into a range. And I don't think that's particularly the best way to go. I have a different take on it. What we prefer to do is train them using a percentage. We say, here's a certain window, and we want a certain percentage of z-scores in that range. Not all of them, but a percentage of them. And you might be surprised to hear how low those percentages go and how small the target windows can get. We can actually work with target windows of 0.8 standard deviations and percentages as low as 50 or 60 or 70%. So rather than trying to force all the outliers into a range, which is one of the criticisms people came up with, is why would you train an outlier? The answer is we never trained an outlier. We don't want to train outliers. We're trying to train the body of the z-scores. And we can also combine this with power training or other types of training. I think I have my little software manager going. Okay, good. So here's a simple example of coherence range training. We're going to give feedback when the z-score for coherence is between plus and minus 1. So what you see down in the graph below, first of all, you see two channels of EEG. You see O1 and O2, and you see the raw EEG. Then below it, you see the z-score for the coherence in alpha, and two green lines, which are the target area, plus and minus 1. So what we can do is we can reward the brain when the coherence is between plus and minus 1 and not otherwise. So this person has low coherence, and you can understand that when that coherence falls between the two green lines, they get a beep or a tone or a movie or a video or something. But if it goes above that range, the feedback also stops. So you don't have this issue of overtraining coherence. You're not trying to go, oh, let's train coherence up and then see where we go. A number of people have reported, John Walker, late Joe Horvat, others, that when you do that, you have to stop and reassess about every 4 or 5 sessions because the brain responds very quickly to coherence information. So if you do want to train coherence into a normal range, this is a very good method to do it and ensures that you're going to be training it into the range, not into just up or down. Get my thing to go down. There we go. So we now can define a range function. And we can go, okay, if the z-score is in the range, we get a 1. And if the z-score is not in the range, we get a 0. So now we can count z-scores. And this was one of the first things we started doing that was pretty different, a little bit innovative, that now you can get a value that if, for example, beta coherence is within 10% of 30, you can get a 1. But now you can apply the range to the z-scores. So now you can score a 1 if the beta coherence z-score is within some range of any target. And the target doesn't have to be 0, but it could be 0. So here's an example of 4 coherent z-scores being trained all at once. And the graph above, again, is the raw EEG, but the graph below is the number of coherence z-scores that are within the target range. And there are 4 z-scores we're looking at, delta, theta, alpha, and beta. So the only way to get a 4 is for all 4 z-scores to get inside their range, and then you get a 4. Then you get your ding. So this was one of our first experiences with training multiple z-scores, actually counting the z-scores. And in this case, we did require all 4 to be within a range. And this was found to be a very useful, a very effective method, because now you're not just trying to train one coherence up or down or within a range. You're taking all 4 of them. And the brain now has a lot less options of how to try and self-regulate. You're basically saying, look, I need you to get 4 of these things, and that's what we're looking for. And so we started using this way back when. One of the first clients we did was done with 4 channel z-scores, even with, or 4 coherences with just 2 channels, and then we moved to 4 later. Now what you can see is the number of z-scores is going to suddenly grow once you make this intuitively. Here's an example of combining z-score training with standard training. And we actually developed this with a client that a couple other folks were participating in. I remember we actually had Barry and Joel Lubar on the phone call when this was being tried. And it combines a lot of elements. It combines the coherence z-scores on the graphs on the left you see. The top one is showing the coherence z-scores. And then the third graph is showing that the coherence z-scores are good and that the power conditions are met. And what we have is we're down training theta and high beta, and we're up training low beta using power training. So what we're able to do now is give feedback when 2 conditions are true. One is that the coherences are normal, but the other is that the power training is being satisfied. And the bottom graph shows a little exponential decay. Every time that person met that condition, they get the movie for a few seconds, and then there's a refractory period. So this really satisfies a lot of the criteria that Barry and others have articulated, the need for well-defined criteria that are met for a fixed period of time. Then you give a discrete reward, followed by a refractory period. There's no reason at all that z-score training or combined z-score and traditional training can't use these great elements, these important elements of operant learning. So we introduced the PzOK. People might have heard about it and wonder, what is PzOK? PzOK is a metric which is the percentage of z-scores that are within the range. And if you have 1 channel, you have 26 z-scores. If you have 2 channels, you have 76 z-scores. With 4 channels, you have 248 z-scores. And the value of PzOK is 0 to 100. It's a measure of how many z-scores are inside the targets. And we have a lot of flexibility here. Now, if you want to get all the z-scores in the target range, then you set your percentage to 100. Nobody we work with does that. Some of the other practitioners are using that idea that I want all the z-scores inside the target range. I'm going to show examples that show that, maybe you don't want to do that. Maybe it makes more sense to just give proportional feedback of how many z-scores, what percentage of them are meeting the condition, and let the brain figure out what to do. So I am more of the persuasion of give the brain valuable information, let it figure out what it wants to do, rather than dictate what the brain has to do because I said so. So our targeting strategy is based on the size of the bullseyes, the number of targets required. We can bias targets up or down. Targets may turn out to be enhances or inhibits depending on where the z-scores are. Are they high or low? Wide targets will automatically select the most deviant scores. And narrower targets will select the body of z-scores in the middle of the distribution. And the training automatically combines and alternates between whether you need amplitude or connectivity training because we're taking all the z-scores into consideration. So you have a lot less decisions to make. The client's brain has a lot more decisions to make, but that's what that's good at. It's good at understanding what you want. So this picture shows conceptually the targets and the z-scores moving in and out. And on the left, we have a narrow target, and we might only require 50 or 60% of the z-scores to get in there in order to get a reward. It works very well. You could use medium-sized targets, in which case you might say, okay, I need 70 or 80% of the z-scores to get inside. And as you see, if you use wide targets, you're going after the outliers. You're typically going to be looking for 90% or more of the z-scores to go inside. We tend to do very little work on the right side. We're rarely going after the outliers. We're typically working in the middle range or actually on the left with narrow targets. And there's a lot of reasons it makes sense that we talk about and show when we do the hands-on workshops. So what happens when you're suddenly training a lot of z-scores is everything gets simple again. Now your feedback variable is the percentage of z-scores that are okay. It's one number, PzOK. And now we're back to where Neil Miller and B.F. Skinner would be very, very happy. We have one metric. We have one criterion. Is it above or below its own threshold, which is the percentage of z-scores I need? And then we have a feedback contingency, and in this case you can see that this is over its threshold about 32% of the time, and so you would be getting 32% feedback, and typically we adjust this so the client's feedback level is around 60, 70, 80%. That's the amount of positive feedback they're getting, and we can do that by adjusting the size of the targets, and we can also do it by adjusting the percentage of z-scores that are necessary. So everything suddenly gets simple again. There's one training variable. We have a couple adjustments underneath it, but we're back to good old operant learning theory, the way it was developed in the 30s and 40s. Now we're giving the brain a lot of information to work with. So, for example, if you change the percentage of z-scores that need to meet criteria by reducing the threshold, that's the green line going down. You see the red line going up, which means it's doing that more often. It's more successful. If, on the other hand, we widen the target window so it's easier and easier to hit the bullseyes, you'll see that you get a higher achieved rate, and you also get a lot less variability because so many z-scores are in there, and that also goes for the most deviant scores. So we tend to want the z-score parameter, PzOK, to be around 60 to 80% or so. And we adjust the target size appropriately, and then we get a feedback rate of about 70, 80, 90%. And so you have a lot of flexibility. You can adjust that where it makes sense for your client. So we now replace the threshold with the target size. And the feedback is determined by the target size and the percentage of hits required. Okay? So no one is driving after the outliers. No one is forcing everybody towards zero. We're providing a lot of information. It kind of eliminates the need for auto-thresholding, where people moving thresholds around. It does integrate QEEG analysis with the training in real time. The protocol automatically and dynamically adapts to what is most needed. We are not designing the protocol. The client's brain is designing it, and I'm going to show examples of that. And it's very consistent with established QEEG-based procedures that have demonstrated efficacy. So in many cases, when you send a QEEG in, the person will simply say there's excess beta on F4, train down beta at F4. Well, Z-scores automatically does that, and it does other things. Now, I'm going to take a little segue into what is actually going on in the brain when this happens. And I'm looking at the work of John Nash, the Nobel Prize-winning economist, the subject of the motion picture, Beautiful Mind. And he described equilibrium points in games in which people are establishing strategies and payments, and they can combine inputs such as bets or decisions. And the individual takes a strategy and tries to maximize their payoff. Well, it appears to me that this is what the brain is doing when you do Z-score training. You have a range of inputs. You have one or more output functions, which is a payoff. The organism will seek optimization and reward. There is an equilibrium which says any change reduces your payoff. And this is the key. I believe that clients who are stuck in defeating behaviors, defeating mental styles, dysregulation, they're stuck in a Nash equilibrium. And any change to them is going to reduce the payoff. So, for example, if little Johnny knows the only way he gets attention is by throwing milk on the floor and yelling and pinching his sister, if he is asked to cease that behavior, his payoff goes down. because he no longer gets the attention. So he is stuck in an equilibrium point. Now, the next important point is that multiple equilibria exist in a complex system. So that one that little Johnny's stuck in is not the only one available to him. He just doesn't know how to get to the other one. Neurofeedback introduces new criteria to the brain in order to seek its dynamic equilibrium. And this is where I think neurofeedback in general has its power, particularly Z-score training. So there's a game in the United Kingdom that's called Brainmaster. And you put pegs in holes, and your tormentor, or your judge, if you will, gives you pegs telling you how well you did, and if you got the right color pegs in the right holes. And you could think of these 4 pegs as being 4 Z-scores. And if you get 4 Z-scores of the right color, you're going to get a ding. And if you get less Z-scores of the right color, you're going to get less dings or no dings. And this game is based on a search strategy where the player is supposed to try and figure out what are the colors they're looking for, what are the colors they want me to get. And the brain, by looking at these, would figure out, for example, by looking at the 4th cell, I have 2 points. And then I look at the one over on the right with 2 points, and the commonality I see is that there's 2 yellows. So the brain would figure out, oh, I can just keep those 2 yellows going, and then I can move from there. We have evidence that the brain is doing this when it's doing Z-score training, and I'll show some data in support of that. Here's one piece of data. This is a naive subject sitting through a 40-minute session, live Z-score training. Happens to have alpha deficits as well as alpha excesses. And at the beginning, you see the Z-scores are as much as 1.8 standard deviations out. And they're kind of bobbling up and down during the initial Z-score training. You actually see them bouncing up and down. I interpret this as that client's brain trying to figure out a strategy. Well, right around minute 12, this client suddenly said, I think I'm getting it. And as you can see at minute 12, 13, 14, 15, 16, so for the next 8 or so minutes, he's doing a really good job holding those Z-scores inside just by letting the dolphin swim. This is a standard, relaxed neurofeedback protocol. He's watching a movie, letting the movie happen. That was his only instructions. Allow the dolphin to swim. And he said, I think I know what it is you want me to do. And we asked him, can you articulate it? He said, no, but I think I know what it is. And there was some talking and things going on in the middle. But for the last 3 minutes, I said, just be quiet, relax, and let the dolphin come. Allow the dolphin to come. And look how those Z-scores packed in there. So for this client, at that time, his brain learned in 40 minutes, and this was his first session. It learned what it is we were looking for and to bring those alpha scores in toward more normal. And it's just a good example that the operant learning, that what we call acquiring the task, actually occurs, and that the operant learning is definitely a mechanism here. Here's the progress of the multivariate variable, the percentage of Z-scores. This is the percentage I'm talking about. And so you see at the beginning of the session, for the first 12 minutes, his brain exhibits what I interpret as hunting behavior. It's changing things around. And on several occasions, you can see it kind of failed. It got worse and went up again, and then it went down again, and then it went up. And then at minute 14, 15, you see he really got it figured out. Suddenly, he was able to get 90% of those in there, and that's when he said, I think I got it. I know what it is you want me to do. Now you see from minute 17 through 40, basically a continuous, steady improvement moving up and up and up. For the last few minutes of the training session, he really does have a large number of those Z-scores in that range. So if that's the mechanism you want, if you're trying to encourage them to move into a given range, there's no question that this technique allows you to do that, and that's what we're seeing. So, important comments about the policy you adopt. EEG deviation should be consistent with the clinical presentation. EEG normalization should be reasonable. It should make sense to normalize it. You want to think about coping and compensatory traits. You want to consider peak performance traits. You do want to think about phenotypes and recommendations. If someone is mixed fast and slow, someone's low amplitude fast, low amplitude slow. What if they have fast posterior alpha? We don't just go blindly in and throw z-scores at everybody. You have to understand what you're doing and why, and you want to monitor subjective and clinical changes. And obviously, it has to be fully integrated in a clinical practice. This is not a get-well-quick, fixed brain machine. You have to know what you're doing. So if you're using live z-scores, what does it make sense to normalize? It makes sense to normalize excessive frontal slowing. Who wouldn't? Excessive beta, excessive high beta, headaches, anxiety. When you see hypercoherence, it might make a lot of sense to normalize it, but not necessarily in the left hemisphere. I've seen people with hypercoherence in the left hemisphere, and they're very functional. They're very happy that way. They speak eloquently. You might see hypocoherence. It might make sense to normalize it, but maybe not. What if it's C3, C4 hypocoherence? I see that in some what we call peak performers, some high-performing individuals. Localized excesses or deficits, it might make infinite sense to normalize those, especially if they're associated with some dysfunction, some dysregulation. But there are also coping and compensating z-scores we have to be aware of. For example, some people have diffuse low alpha. I have a couple clients like that. It could be that it's a chronic pain situation and that low alpha is a coping mechanism, a barrier to perceiving the pain. Sometimes you see diffuse high alpha, and that can show up in cases of chronic anxiety. Again, a coping mechanism. Posterior asymmetries may exist, PTSD, stress coping, cognitive dissonance, these types of things. Substance abuse or addiction. A client may show mixed fast and slow, or they may show excess alpha as a way of coping, compensating for their problem. There are peak performance z-scores, things you see in high performers, such as this left hemispheric hypercoherence, central intrahemispheric hypocoherence. Excess SMR, this is one of my favorites. When I see excess SMR, I ask questions. It may not be a problem at all. Excess posterior alpha may not be a problem at all. Fast posterior alpha may not be a problem at all, but you have to ask. If a client with fast posterior alpha reports that he or she is uncomfortable, they're buzzing, they can't stop thinking, their thoughts are racing, then that the fast alpha may be associated with that. But if they say, I'm happy, I'm doing fine, I can do a lot of different things, I'm very effective, I'm not anxious, then you're not going to particularly want to go after it. And you can avoid normalizing anything simply by keeping the EEG sensors away from those areas. And in cases of head injury, for example, you may want to keep the sensors away from an area that's holding up the rest of the brain, that's compensating. I mentioned phenotypes. I'm not going to go into vast detail, but I will acknowledge Jack Johnstone and Joy at Lunt and Jay for articulating this. Martain also, Arns has done a lot of nice work. A lot of the phenotypes are going to show up as z-scores. Your diffuse slow, focal abnormalities, mixed fast and slow, et cetera, et cetera. Some of them won't show up as z-scores. Epileptiform may not show up in a QEEG. Fast alpha may show up in the wrong way. The z-score may think that there's excess alpha or excess beta, no less, if it's fast. So you have to look at the waveforms. You have to look at what's going on underneath and make a decision. Many phenotypes can be addressed with z-score training. A lot of the phenotype recommendations basically say to normalize the deviation, and z-scores can do that. You could also add other training. If the phenotype recommendation, for example, says, uptrain alpha, then you can add that. And phenotypes don't currently look at connectivity, while the z-scores can do. So that's one difference between those approaches. But to me, they're very compatible. They're consistent. I always try and understand if there's a phenotype here and how to work with it.
I'd like to interrupt here, Tom, if I may, I'd like to interrupt here for a second. I want to give the validation statement, which is honeybees communicate by dancing. And to let you know that you've got about 10 more minutes of lecture. And if anybody has any questions, they can write them into the chat box. Thanks.
Thank you, Cindy. Okay. So this was the article that was in the Budzinski book a few years ago. And myself, Bob Thatcher, Mark Smith, Bill Lambos, and Dick Stark reported on this technique and reported on some interesting cases. One of them was reported by Mark Smith of a 3-year-old male who had a concussive head injury. And he had seizures, and they had been trying inhibit training and coherence training and kind of searching around. And then he switched to Z-scores, the all coherence is normal that I talked about, and they had a dramatic improvement. And this young man is now, right, like 11 years old, and been tapering off the meds, but has not had another seizure, which is a good thing. And here you see their cue before and after a conventional training approach, where they just went after some amplitudes and went after a coherence using a regular vanilla neurofeedback package, and there was an abreaction here. This actually caused problems. And so what you can see is, even though the training did well address the power issues up on top, the delta excess and the beta excess were well addressed, you see the emergence of a lot of coherence problems down in the third connectivity, the fourth row, which is the middle connectivity row. Because the training did not incorporate coherence, and certainly not coherence Z-scores, the brain responded by going, yeah, I'll take care of these power abnormalities and watch what happens. I'm going to do this to the coherence because you're not telling me otherwise. And so this was a less than optimal approach. Using Z-score training really went after those coherences. So now if you compare left to right, you'll see that the Z-score training, using a global criterion, the brain actually elevated alpha a little bit, but really went after those coherence abnormalities, and the coherences were cleared up. And interestingly, some phase things popped in. So we're letting the brain decide what to normalize, because certain things are left out of the percentage, and we don't pick them. Again, I keep emphasizing that the brain is designing the protocol. So this was a good outcome, one of our very, very first. Here's another case of a 7-year-old male, ADHD, and used PzOK training, and we just used F3, F4, P3, P4, and then went to F3, F4, C3, C4. And this kid was very belligerent, very defiant, hard to work with, a lot of anger. And you can definitely see the delta excess, and these were artifacted. This is not eye activity. You also see that posterior cingulate in delta. And again, massive coherence excesses all over the place. And on the right is, after 21 sessions, the Z-score training. So the Z-scores really go after the variety of things that are a problem. And this is just 4 channels of Z-scores. And there was very good clinical benefit here. The kid really changed his way of thinking, feeling, and behaving. Here's Loretta validation, showing that even though we only trained 4 channels, the Loretta Z-score is normalized. So on the top left, we have 3 standard deviations of abnormality, and on the bottom right, it's 0.4 standard deviations. So 4 channels is really sufficient to get a grip on things going on inside. It's just that I think Loretta training is somewhat faster, more specific, but we still do a lot of 4-channel work. Here's his eyes closed pre and post. He was only trained eyes open, but we did the cue eyes closed. And again, all these abnormalities which were evident eyes closed were resolved just with the eyes open training. Again, here's the Loretta. Here's his posterior cingulate. On the upper left, it's 5 1/4 standard deviations. On the lower right, it's 1.2 standard deviations. So again, good normalization of Loretta just using F3, F4, P3, P4. We then went and published a set of case studies. These were my co-authors, and we also had other contributions from other people, Nancy Wigton, Dorothy Klein. Other people had contributed. They only wanted 5 authors, so they got the top 5. And here's an example of the Z-score changes in 10 sessions. So these are 3 cases, and as you can see on session 1, they had anywhere from 25% of the Z-scores to 45% of them outside 1.0. And after 10 sessions, it was between 10 and 17, 18% of the Z-scores that were outside of that range. So the Z-scores actually learn to move in. They're learning to go inside their target ranges. Here's another example. Interestingly enough, one client whose connectivity Z-scores were, 75% of them were outside, he got it to where less than 30% were outside. The pink client is interesting because his connectivity Z-scores actually, technically, if you will, got worse. from 10% to 11%. But what happened is other things normalized. So even though some coherence z-scores were allowed to move outside the target ranges, other things normalized, and there was a clinical benefit there. Here's another example of this is between session 1 and session 10. The blue dots is the first session. As you can see, there are z-scores outside all these different ranges. 10% of them are outside 2.5. 10% are outside 3. 5% of them are outside 3.5. They're all over the place. And after 10 sessions, you see this curve with the pink dots, which really shows they pulled in. Most of the z-scores now are within 1 or 1.5. And a very small number of them are outside 2, 2.5, 3, 3.5, 4. So visually, you can really envision that the z-scores are pulling. Here's another example, similar kind of a thing, a very dramatic, where the blue is the pre and the pink is the post. The z-scores are really learning to get inside those ranges and stay there. I'm going to get close to where I can break and save some of the future stuff I have for another workshop, more advanced topics. But this is a very interesting case of an ASD. And you can see on the left a lot of deviations. You have a deficit of delta power. You have deficits of theta and alpha. You have excess high beta. You have a lot of hypocoherence, looking at the fourth row. You have a lot of excess phase. And on the right middle, let's say Q2, is after 20 sessions. You see a lot of normalization. You even see some of what we call bounce. Things are moving in the other direction. There was good clinical improvement at that point. Again, we're not trying to force everything towards zero. We're allowing the brain to make decisions what it's going to move towards zero and what it wants to leave out. And you look at the extreme right, and you'll see then a lot, again, more normalization. There's a little bit of other things emerging, which are fine with us. You know, you see a little bit of excess delta relative in the back. But look at how the connectivities have pretty much cleared up. And, you know, the powers, if you look at the bird's eye view, again, very much normalized. And there was dramatic clinical improvement with this young kid. And, again, this was 4 channels of live z-score training. Here's an example just showing the difference between the live and the static maps. They're very, very similar. The maps on top were done with NeuroGuide using the ANI database. The maps below were done with BrainDX using the New York University database. And there's some difference in the exact size of the particular z-scores, but the basic findings are the same. The locations where there are excesses or deficits are the same. The relative degree of excess or deficits are the same. So we're really confident that we're getting good information even as we move now from the ANI to the New York BrainDX database. The live z-score training is going to have very, very similar functionality, and it's probably going to be a very smooth transition from one database into the other. Couple advanced things we're looking at here is z-bars. Z-bars are colored bars showing you the averages of the z-scores and little tick marks and lines showing you their instantaneous values. Now here's the exciting, interesting point here. The z-scores that are most normal are also the most variable. You see the variations largest around the little white squares, which means they are normal. What it tells you is that the way these z-scores get normal is by having variability, by bouncing around, by having flexibility. Nobody is ever stuck at zero. That doesn't even make sense. But if you look at the 2 blue bars, the dark blue and the light blue, they have no variability. They're stuck where they are. So that particular value is stuck at -2.5 standard deviations. And the other one is stuck around -1.5. What we find is when you teach z-score training, you increase variability. You increase flexibility. It's not just moving something towards zero. It's telling it where it is and when it is and allowing the brain to gain flexibility. So, Cindy, should we move now? I see we have just about 15 minutes. Do you want me to do a couple more slides, or should we let the group start to get involved?
Well, I don't see that anybody's got any questions. If you do,
I'll keep going. I have about 10 or 15 more slides.
It looks to me like people would want you to keep going. Otherwise,
okay. Well, all right.
Yeah, keep going, because I
We
see
have
more
something,
questions.
I'm happy to. We have something new called Z+. And Z+ is a way to evaluate the outliers. The Z-scores which are outside the target range and remain outside the target range, nominally, there's no feedback associated with them. And we viewed that as something that probably needed to be addressed. So what we did is we created two new metrics. One of them is called PzMo, which is PZ momentum. We actually have calculated the momentum of the outlying Z-scores as if they were planets in a solar system. And as those outliers move toward the center, the variable PzMo jumps up. And that's what you're seeing on the middle graph. You're seeing the indications when Z-scores begin to move toward normal, move toward the target range, rather. And that gives an additional level of reward. It's kind of a gold star. You're kind of giving the brain a reward for moving in the right direction. And on the bottom, then, we have what we call PzMe, which is PZ mean. It's the mean distance of the Z-scores. So you're kind of measuring the size of the planets, the size of the solar system. And so now you can see that as the brain gets more and more meeting the condition, the criteria, this PzMe variable goes up. So we're adding on top of the basic idea of these percentages of Z-scores, now these new metrics we call Z+, and the basic metrics are PzMo and PzMe, which are PZ motion or PZ momentum, and PZ mean. And a lot of clinicians are using these as additional feedback. Now I'll talk about the use of some of the advanced techniques. We have developed at Brainmaster a live S-Loretta projector, which uses our own software. It's 100% Brainmaster software. We're not talking about downloading Loretta or using anybody else's software. And we work closely with Roberto Pascal Marquis and New York University in developing this system. But we have our own capability that runs live. It runs at real time. Our Loretta projection is about 200 times faster than other techniques due to some proprietary software stuff we've conjured up. So we can actually look at Loretta movies live, and we can also do Loretta Z-scores live, because with New York we now have an S-Loretta database built around what used to be NX-Link, but which is now BrainDX. So, for example, we could be looking at a region of interest, and here we're looking at the cingulate gyrus. And the cingulate gyrus could be reflected as Z-scores. And for every single voxel, we compute a Z-score. Now, some people think that when we do this, we're actually putting Z-scores on the surface and trying to project them with Loretta. That's not what we're doing. We're creating the current source density estimate for every voxel, and then that current source density, we have a Z-score for it. So we know the normal ranges of current source density for every one of the 6,239 voxels. So in this example, you would see the anterior cingulate, the frontal areas are a little high. And this could be beta, for example. So the frontal areas are a little high. The middle of the cingulate gyrus is normal. Green is normal. I think of it as green jello. Lime jello is good. Cherry jello is not. And in the back, you see the posterior cingulate area also has a little bit of an excess. So by using Loretta, you get extremely detailed localization information that you'll never see on the surface. Now, one of the drawbacks is you have to use 19 channels to get the S-Loretta. But if you're going to cap people up and do 19 channels, this is definitely one way to get very detailed, very fast information. And people are working with chronic pain, they're working with anxiety, and the clinicians who are working with direct Loretta training say that it's very fast, and the client has a very profound sense of what's going on. In fact, you can use this as a display. You can train right on this and say, make it green, make green jello. And it can be surprising. Clients in 3, 4, 5, 6 minutes can get the hang of what it is, and now they're quieting down that cingulate. Very hard to do that on the surface. Here's an example of a lesion which was imaged using our live S-Loretta. This individual has a degenerative lesion in the mesial temporal insular area, visible on an MRI, and we visualize the same thing on S-Loretta using Z-scores. So very powerful method, and you wonder why anyone would be doing fMRI and SPECT and PET and all this. You can get EEG recordings with almost similar resolution and much, much faster and a little bit cheaper than a couple million. Here's an example of live S-Loretta z-scores used to image that lesion. And if you look at the z-score display on the left, the range, you'll see that I'm starting at minus 2.9 standard deviations and going down to minus 4.8 standard deviations. So we are selectively imaging only those voxels whose activity is 3 standard deviations below normal. So we are not actually imaging activity, we are imaging the absence of the activity. We're imaging the lack of activity. And that was very profound, very exciting to realize you can actually localize an area that is electrically silent by using Loretta z-scores. Here's an example of some more live z-scores. This is using the BrainDX database. This is the type of display you get now if you're using Brain Avatar with BrainDX. These are surface maps. They are functionally equivalent to NeuroGuide maps or NX-Link maps, but they're live and they're done in real time and they can be used for training. So you could be training directly off these screens or training the derivative variables thereof. Here's another example of a Loretta z-score display of gamma. This was an individual who was a very interesting kind of a person, very intuitive, very tuned in, and had a really profound gamma. So I thought it would be interesting to image it. And what we're finding out is that it's in the mesial areas, more centralized. I mean, it's associated with area 17. But what you're looking at is the right hemisphere, and you're looking at the mesial surface, the inside surface of the right hemisphere. Very interesting to see that we can spot that gamma internally and determine, where it is in terms of these deeper areas of area 17, having to do with visual and other types of experiences. So as a summary, we have new methods that use normative data. It is a comprehensive whole head approach. We can normalize activation and connectivity. We use multiple targeting and biasing capabilities. It's consistent with QEEG and phenotype approaches. We provide the brain with complex information and let it understand what to do. It is a very simple training format. The brain is not trying to figure out 100 things at once. It's basically just trying to figure out where do they want me to go. It's effective for assessment. It is effective for training. Here's a reference. We look at the position paper on QEEG by Hammond and Walker and all these other good people. And we believe that z-score training as we do it is very consistent with these standards because it is a QEEG-based method. And there's also a handful of other references I have here. And I acknowledge Cindy and Jay and I did an article way back on combining phenotype and z-score modalities. That was a very fruitful dialogue. We point to Johnstone, Gunkelman, and Lunt on phenotypes. And of course, I have to acknowledge Berger and Kaiser and Mann and Suyanobu on the simulated visual motor task. Berry's paper on physiological origins. Silverstein's paper on dynamic sculpting using inhibition. And Mark Smith, Dick Stark, Bob Thatcher have published some nice articles in Neural Connection. And I look at John Walker and Jerry Kozlowski and Bob Lawson's paper on modular activation coherence. We're consistent with all of this literature. And so to us, z-score training is really a very flexible and scientifically validated way to go after neurofeedback. So
Tom,
that's the end of it.
We have a question from one of our attendees.
Sure.
I've used 2 live S-Loretta maps simultaneously for a region of interest, each at a specific frequency band, looking at cross-frequency coupling and training. Anyone else using this clinical approach?
Well, yeah, we have a handful of clinicians who are doing it. I will emphasize that we provide tools, and we encourage people to be ambitious and creative. So I know Mark Smith is doing it with chronic pain. I know of a clinician whose name is escaping me in the Colorado area who's working with very difficult clients. Penny Jean does it. If you look at the winter neural connections, you'll see a number of articles on people doing multiple S-Loretta training, and that entire neural connections doesn't even talk about Z-score training. It just talks about S-Loretta power training, and we can get S-Loretta connectivity training. And yeah, I mean, I would send people to that neural connections, winter neural connections. There's also stuff on our website and our knowledge base posted on that regard. So absolutely, there's a growing body of people doing it.
Okay, I don't see any more questions, so if you wanted to continue, feel free. We've got a few more minutes.
Well, I think I'm done. Aren't we about wrapped?
Yeah, absolutely. All right, then, sure, there's no more questions, anyone? Type away. This is your last chance.
I will let Cindy get a plug-in. We've been asked to do a second one of these, and we're going to do it on advanced stuff. I'm going to talk a lot about brain DX, a lot about S-Loretta, and a lot about some other approaches towards Z-score training.
Yeah, and we set that date for May 9th. So look for that if you're interested.
Got it.
Yeah. Okay, so I'm going to repeat the validation statement, which is honeybees communicate by dancing. And then, when we have a thank you from one of the attendees who says that she's learned a lot and now I need to assimilate it. Yes, as Barry Sterman would say, you need to go sit with your eyes closed and let it consolidate.
Amen.
Yeah.